Almost.
Because I need to put some.
You can go ahead and do that while we just sort of make sure the picture looks good. I think over there, Helen.
There's nothing incriminating. I don't have any.
I think there'll be plenty. Autumn Lynn has plenty of that.
That's what I heard.
All right. I might have to put it over there. Well, I mean, I could put it over here as long as I have power. That cord's super long. It doesn't necessarily have to be. Hang on. I wonder, you think if I find it on I don't know.
All right.
Hi, that was amazing. Oh, thank you.
Exciting.
Can we test the actual picture? Make sure that works.
Oh, that was
Is that turning on?
Seems like you do one of these every month now.
This week has been something that I've had 2 proposal defenses, 1 comprehensive exam.
I guess it's a good thing. Say you look at the side.
Okay. You ready? Okay. Now all of you in the back can we have the lights out, and so now you can fall asleep. No, you can't fall asleep. Anyway, it's a real pleasure today to be able to introduce Melinda Fowler, who has been here.
Save that for the roast.
You know, she's been here for a while. Well, all right, I'm not going to say that. It's not the point. Anyway, Mel has a really unusual academic history. Some of you heard this before. She did her undergraduate work at University of Long Island, Southampton College with Maria Kretzmann, who was a graduate student here with me quite a few years ago. And so she applied to come out to California, but being an out-of-state student was kind of difficult. So she then took a detour to Sonoma State and worked with Dan Crocker, who was also a graduate student here. So she did her master's with Dan Crocker actually she worked on female elephant seals. But in a pinch, I was throwing this together we had an earlier exam this afternoon. So she worked on northern elephant seals, some aspects of physiology. And then she sort of came on the Sonoma State UCSC corridor and didn't stay at Año Nuevo. She just kept coming down in here, where she worked on elephant seals again. But she started with the idea that she was going to take. Great. Oh, I didn't. It's not. I did the arrow. Your computers. What am I not doing on here?
I don't know.
It's not a projector. All right, well, maybe I'll just do the.
Wait. Here we go. There we go.
There. Okay. So somewhere along the line between going from Sonoma to here, she decided to do a little bit of field work. She spent 4 months. Twice working on monk seals on not this island, but it was probably not much bigger than this island. So after that, I figured, heck, anybody that cannot kill the other person and work on monk seals on a little island for 4 months has got to have something right. She came here, and amongst the many things that she did, she actually was going to try and do some of the physiological measurements and interactions that we do with elephant seals on California sea lions, or one of the odoriids. And she led several of our field projects down on San Nicolas Island, and then also went and did some work in the Galapagos, on Galapagos sea lions, working with a colleague from Germany, where she also should have been able, I can't remember if you actually got your hands on it first. Yeah, she did Galapagos fur seals. And then she, more recently, normally I give these talks about where everybody in my lab's traveled. I'm giving a talk on all the animals that Melinda's worked on while in my lab or associate. She spent, and this one I'm really jealous of, she spent a season working on emperor penguins with Paul Panganis at Cape Washington. And I took this picture, I got to spend a day, she got to spend several months. She also got out to take a little quick visit when we were down there working on Weddell seals. So she actually, even though she wasn't on a Weddell seal project, she got to play with Weddell seals out in the Antarctic. But then came back to UC Santa Cruz to do what many people in my lab do, because it's such a wonderful system to work with, and that's to work with northern elephant seals. That's the talk she's going to give to you today. And just to let you know how I've given you a diversity of the animals she's worked with, she's going to leave here soon and go up to Victoria, to Simon Fraser University, working with Tony Williams to work on this amazing this terrestrial bird, but doing, again, the interaction between physiology and behavior. So I will stop there and let Melinda figure out her computer.
Thank you. Okay, cool. I think it's working. So thank you, everyone, for coming. It's quite a full house. This is great. I am going to tell you today about some what I think are really awesome physiological characteristics of elephant seals. And so we could get started, and a simpler talk for my title could just be Seals, Fat, and Milk. Those are the basics of what I'm going to talk about today. As far as an outline, I will first tell you a little bit about elephant seal life history. A lot of you are pretty familiar with it already, but I'm going to explain some of the really amazing things they do, both ecologically, and then I'm going to try and kind of link that up with some of the physiological adaptations that I've been studying for my thesis. And so that will require a little bit of a review of some physiological mechanisms of lipid mobilization. So we'll kind of review that so we can, hopefully you'll be able to follow along some of the more intricate biochemical slides. And then I'll go into each one of the chapters of my thesis, and each one it will be investigating a specific aspect of lipid mobilization and most of it in elephant seals. So first, to kind of back up a little bit, lots of different animals undergo periods of fasting in their lives. So it can be just short-term between bouts of foraging or the time it takes to get to another foraging location, or it can be longer-term, where it's sort of a part of their life history where they spend time, a long period of time without ingesting any food. And often those times are coinciding with depressed metabolic demand. So animals will go into hibernation or torpor, where they don't require as many calories to function. But elephant seals take this to the extreme. So they spend several months at a time abstaining from food, but they don't do the depressed metabolic demand thing. They actually do all kinds of crazy things that require a lot of calories, while they're fasting and not eating anything. So one of the things that really interests me about physiology is some of the physiological adaptations that enable animals to occupy a particular niche or do a particular thing. So I'm interested in what sort of physiological mechanisms can make these strategies possible. So they're able to go and forage far out at sea and then fast on land. How do they actually do that? So, like I mentioned, elephant seals spend a lot of time foraging out at sea. In fact, the majority of their life, they're out thousands of kilometers in the North Pacific. So they'll go to sea for several months at a time, and they're exploiting a food resource that's so far from land, but they're still tied to land for a couple of different fundamental life history stages. So they have to come to land to breed and to molt. So whenever they're on land, because their food resource is so far away, they can't do the commute back and forth. So they got to go out to sea, forage, accumulate a bunch of stored reserves, and then go back to land and take care of their business on land, but they can't eat anything on land because their food resource is so far away. So as I mentioned, elephant seals don't do that depressed metabolic demand thing. They just go to the opposite extreme and do all kinds of energetically expensive things while fasting. So the males during the breeding season spend a couple of months kicking the crap out of one another and don't eat anything. The females have a pup and nurse the pup for a month without eating anything. Also during the molting period, they come ashore and shed all of their skin and fur and grow all new skin and fur without eating anything. And additionally, they have this post-weaning developmental stage where the pup has been weaned and it's actually developing while fasting, which is also this really interesting, rare characteristic. One of the things that I'm super interested in is this rare combination of fasting and lactating. So that's super rare to combine fasting and lactation. Lactation is a critical aspect of provisioning the young in mammals, and it's really expensive. So most mammals dramatically increase how much food they ingest during lactation. But elephant seals are doing this fasting at the same time, so they've got to ration the fuel resources that they have, but at the same time, they're creating this food for the offspring and shunting huge amounts of calories to the offspring, but they've got to do it only on stored reserves. So this is a really rare combination. So if an animal wants to fast and do it for a very long time, the best way to fuel this is through fat. So lipid is a great storage molecule. You can store it for a long time without a whole lot of water. You don't have to spend a lot of calories to maintain it. And energetically, it's the most bang for your buck. So per gram of tissue, you get the most energy out of it. So consequently, elephant seals are really fat. They're good at utilizing lipid. They power the vast majority, about 90% of their metabolism when they're on shore from lipid. So the adult females, they show up, they're very, very plump, and they utilize a huge amount of their lipid stores and shrink while making this pup very large. So they spend a lot of their time out foraging, accumulating this lipid mass, and then they come to shore and they have a pup, and over the course of about a month, 26 days, they feed that pup, and it gets enormous. And we're not talking just maintenance of this pup, we're talking gigantic, tripling in size from like 20 kilos up to over 100 kilos. And the fact that this pup gets so fat is important to the pup for the next stage in its life. So the pup, once mom has given all the resources she's got, she weans the pup and heads back out to sea. And the pup is left on shore for 2 to 3 months to develop its diving capabilities. So once that pup leaves, it forages like an adult, and it is independent and has to figure out what to do all on its own. And it requires the physiological diving capabilities in order to do this. And while that pup is on shore, it's developing those diving capabilities. So it can't leave and be an efficient forager without having developed those stores. So it's important to be fat enough to be able to develop the diving capabilities. So it's really important for the pup to be fat so it can survive in the next stage of its life. So how does mom do this? How does mom make that pup so fat? She puts tons of fat into the milk. So this figure, we've got days after birth, and this is the percent lipid composition in the milk. And this is about day 5. This is up around 30% fat, 5 days after birth. And then if you measure it again, take another sample about day 22, it's up around 55% fat in the milk. That is a lot of fat. If you think about whole milk's about 4% fat. So this is almost like pudding at the end of lactation. So I was really interested in how does all that fat get in the milk? So it's really important for mom to be able to transfer this fat to the pup. And so how does that work? If an obese human were to give birth and suddenly stop eating and lactate, they would not make 55% lipid. So there's something different about elephant seals. How do they do that? And I also started thinking, how does mom ration her fuel reserve? So she's got to make all this fat-rich milk at the same time as rationing her own lipid reserve. So how does she manage all of these things? So those were two of the kind of big, broad questions that I started thinking about. So elephant seals have blubber layers, and so we're going to zoom in a little bit. And in this blubber layer, there are fat cells, so adipocyte cells. And in this adipocyte, there's a triglyceride molecule. And you've basically got 3 fatty acids attached to this backbone, and this is the storage molecule. So I'm just going to call it TAG from now on. It's got 3 fatty acids, and it's in the blubber, it's in the adipose tissue. But then, in order for the rest of the tissues to utilize it, it has to get out of the blubber and into the circulatory system. So these fatty acids have to be removed from the adipose tissue and get into the blood vessel. And then from there, they can be picked up by other tissues, such as the mammary gland, to create milk. And hormones and enzymes can affect these processes. So I'm going to go over a little bit of biochemistry background here, and I'm going to talk a little bit about some of the enzymes that facilitate the release of these fatty acids. So enzymes that can cut up fat and release them from the cell. One of them is hormone-sensitive lipase. I'm going to try to have the little blue triangle from now on for hormone-sensitive lipase. And another one is adipose triglyceride lipase, which I'll probably call ATGL because it's less than a mouthful. And these two enzymes, their job is to turn that TAG molecule into free fatty acids. All right, so they cut up those free fatty acids, and now you've got free fatty acids in the circulatory system that can be utilized for other tissues. There's one other enzyme that I want to introduce you to, and that is one that will grab fat from the circulatory system and bring it inside a tissue. And that's called lipoprotein lipase. So lipoprotein lipase will be on the outside of a tissue and can grab circulating lipids from the bloodstream to bring into a tissue. But the thing about lipoprotein lipase is it only grabs TAG molecules. And you're like, well, didn't we just cut those up and put them in the bloodstream? Well, sometimes some of these excess free fatty acids will be repackaged into TAG molecules. So now you've got repackaged TAG molecules and free fatty acid molecules floating around in the bloodstream in order to be picked up by other tissues. But lipoprotein lipase is required to pick these up, this TAG molecule in the bloodstream. So put this in kind of a bigger picture, like kind of put the whole story together. We've got a big fat mama elephant seal, and she's got a bunch of blubber stores, and she's got a bunch of adipocytes. And in order to release this TAG from her stored blubber, she has these enzymes that cut up fat. They're lipolytic enzymes. And that releases the free fatty acids into the circulatory system. Hormones can affect this process. And then we've got some of these free fatty acids being repackaged into triglycerides in the bloodstream. So we've got these 2 different kinds of lipid metabolites floating around in the bloodstream to be picked up by tissues such as the mammary gland. So the mammary gland is going to pick those tissues up, and here, this LPL on the mammary gland can grab the triglycerides, that TAG molecule, and bring it inside to the mammary gland to create milk. But free fatty acids can come in by diffusion or by small transport proteins. So there's different ways that these lipids can get into the mammary gland. And then the mammary gland, its ultimate job is to take all of that, package it into fluid called milk, which will then make Junior very fat. Now, the interesting thing here is this LPL molecule. I kind of think of it as the doorkeeper to getting these triglycerides into the mammary gland. And there have been studies that have looked at LPL in seal mammary tissue before. So 2 different seal species, they found that LPL increased across lactation and was mirrored by this increase in milk lipid. So they made the argument that LPL is regulating how much lipid gets into the mammary gland. Well, then there was a study in elephant seals, and they measured the same thing, but they found really low LPL levels, and that it was not related to milk lipid content. So then I started thinking, well, if LPL is not regulating how much lipid gets into the milk, possibly the regulation could be on the side of the supply. So possibly just the amount that's available to the mammary gland might be their kind of regulatory aspect for milk lipid content. So for chapter 1, I'm going to talk about the specific fatty acids that are in the blubber and end up in the milk. For chapter 2, I'm going to talk about the hormones that can affect these processes and how they're related to milk lipid content. And for chapter 3, I'm going to talk about these enzymes, hormone-sensitive lipase and adipose triglyceride lipase, which actually no one has, well, when I first started this, no one had ever measured these in a seal before. And there's one study that has sort of started to measure some of these, but not in any lactating species. So that was kind of the exciting new thing. All right, so chapter 1, we're talking about free fatty acids. So we're all pretty familiar with this at this point. We've got an adipocyte, we've got our tag molecule, we've got free fatty acids. They've got to be released, come out into the bloodstream, to be picked up by the mammary gland to make milk. But you've got to remember that mom also has to power all the rest of her metabolism with these free fatty acids as well. So all of her non-mammary tissues need fuel. So some of those free fatty acids will be picked up by other tissues, and a lot of the rest of them will be picked up by the mammary gland to make milk. Now, all of these fatty acids are not necessarily created equal. There are different types of fatty acids that can be released into the bloodstream. So you can have saturated fatty acids, or mono or polyunsaturated fatty acids. And you've probably heard of these. People, they seem to be kind of an interesting topic in nutrition these days. But I'll go over what this means so you can kind of follow along a little easier. It's a little reminder. So saturated fatty acids have no double bonds. It's just a chain of carbons, and it's a straight chain. And these become solid quickly at low temperatures. So think about butter or saturated fatty acid. It's a lot of saturated fatty acid here. It's just room temperature. It's a block. And then we've got monounsaturated fatty acids. And these have one double bond. And this causes a kink in this chain here. And for the purposes of this talk, I'm going to separate them into medium chain and long chain. And you don't have to remember the numbers, but I'll be talking about the shorter ones being the medium chain and the longer ones as long chain MUFA. So when I'm talking about MUFA, I'm talking about monounsaturated fatty acids. These will be fluid at lower temperatures. So think about olive oil. Olive oil has a lot of MUFA in it. And you think about how butter and olive oil side by side. Olive oil is liquid, butter can be solid. And then our last class are polyunsaturated fatty acids, or PUFAs. And these have more than one double bond. And these are more easily mobilized. They're abundant in marine systems, but kind of less abundant overall. They can be important for a variety of things, not necessarily just fuel, but also as signaling molecules. They can be very important to developing neonates. And these are also fluid at low temperatures, and you can think of these as fish oil. All right, so these are our categories of fatty acids that I'm going to be talking about. So I started thinking, are certain fatty acids mobilized differently for milk production? So all these fatty acids are different ones mobilized from the lipid relative to others. In previous studies on other species, they found that the actual structure of these fatty acids matters. So the chain length, so the more carbons there are, cause it to be mobilized less quickly in other species, while the amount of double bonds in the chain can cause it to be mobilized more quickly. Right? So this has been shown in other species, and I wondered, is that the case with elephant seals? I was also interested in which of those fatty acids end up in milk. So I was wondering, is there selective uptake of these fatty acids by the mammary gland? And so which ones are mom using and which ones end up in milk? So in order to do that, we went to Año Nuevo, and we caught a bunch of elephant seals. And early in lactation, about day 5, we captured elephant seals. And then again, in late lactation, we recaptured them, so about 22 days after birth. And we took a milk sample and a blubber sample, and we took a mass measurement, as well as a body composition measurement where we can basically calculate how much adipose tissue the entire seal has in it. So this whole seal, how much of her is adipose stores. And for the lab portion of this, my committee member, Kathy Debier, who unfortunately can't be here, she works in Belgium, and she did the lab work and quantified the different fatty acids in each of these samples. So in the blubber and in the milk, we got about 20 different fatty acids, and we quantified the abundance of each one of those in the blubber and the milk at early and late lactation. And in order to calculate how much was mobilized, I basically took the amount that was in the blubber to begin with, and then the amount that was in the blubber at the end, and basically the difference between them, it's no longer in the blubber, it was mobilized. So I was able to calculate a mobilization index for the different fatty acids, and I grouped them into their classes, like I discussed earlier. There's 20 of these, but I grouped them into their different categories, and I looked at their mobilization. And what I found is that my results matched very closely with previous studies on other animals where the structure did matter, and that the most mobilized were the PUFAs, and then followed by the saturated fatty acids and the medium chain MUFAs. So here's our PUFA, saturated fatty acids, medium chain MUFA, and the least mobilized were our long chain MUFA. And all of this matched the predictions based on this biochemical structure. So that was a really interesting result, that elephant seals are mobilizing lipid, these specific fatty acids, similar to other animals. And then I wanted to look at the milk. So in this figure, we've got early and late lactation. This is the proportion each of these classes is making up of the milk sample. And so we've got medium chain MUFA, long chain PUFA, and then saturated fatty acids. And what I noticed is that overall they're quite similar. Overall, it's very similar between blubber and milk. But there is one category that increases across lactation. So we've got this long chain MUFA is the only category that increases in milk from early to late lactation. But if you remember, the long chain MUFA were the least mobilized of all the fatty acids. So the most mobilized were PUFA, so this is our order over here, and long chain MUFA were the least mobilized. So we've got an increase in the milk of the particular fatty acid that was the least mobilized from the blubber. And so I started thinking, what is it about long chain MUFA that might be beneficial for the pup? Is there a particular reason that an elephant seal might want to put long chain MUFA into the milk? So I started thinking about what could be benefits for the pup. And again, they've got to have fuel for this post-weaning fast at 2 to 3 months. But again, fatty acids do lots of other things. So they serve as these essential nutrients for development. And additionally, they have to prepare this pup to enter this very cold environment. So they're about to go diving in very cold water. So they've got this thermal regulation need as well. So this thermoregulatory layer, and different fatty acids can contribute differently to the thermoregulatory capabilities of the blubber. So I started thinking, what could long chain MUFA be beneficial for? But when we're talking about thermoregulatory benefit, it's been shown in other species, and in fact in the adult females, they accumulate medium chain MUFA on the outside of the blubber. So the area closest to the cold water, they actually deposit high levels of medium chain MUFA. And that's called homeoviscous adaptation. And so that makes sense, but elephant seal pups are getting lots and lots of these medium chain MUFAs. And again, the ones we're interested in are the long chain MUFAs, and they're not as important for this thermoregulatory advantage. So it didn't seem to be something that was really important for thermoregulation for this long chain MUFA. And then I thought about it being essential nutrients. But PUFA are our most important for neural development for pups. And the pups are already getting incredible amounts of PUFA relative to terrestrial animals and the recommended daily allowance for a growing human. They're getting way in excess of that. So they're not really limited on PUFA. And so the long chain MUFA shouldn't be really affecting neural development either. So basically, I wasn't able to identify any kind of definitive pattern for the pup. Like, oh, the pup needs this long chain MUFA to do something. So then I started thinking, what else could be happening? All right, so we've got our mama seal, she's got this blubber layer, these fatty acids are liberated, they go into the bloodstream, they're picked up by the mammary gland or by other tissues. So all of mom's other tissues have to utilize fatty acids. And just like the mobilization can be dependent on the structure of the fatty acid, the utilization of those fatty acids can also be dependent on the structure of the fatty acids. So long-chain MUFAs are also oxidized slower than other fatty acids. So if mom's tissues are oxidizing our PUFA and our saturated fatty acids and our medium-chain MUFA faster than the long-chain MUFA, that will leave a higher level of those long-chain MUFA available to the mammary gland. And so then we would see an increase in this long-chain MUFA over lactation. So I think it's a difference in the mom's other tissues utilization, which causes the availability of these long-chain MUFAs to the mammary gland, which causes this increase. So our take-home messages for chapter 1 is that seals mobilize specific fatty acids similar to studies in other mammals and birds, and that long-chain MUFA, the least mobilized class, is the only class that increases in the milk across lactation, and that this increased long-chain MUFA in the milk is likely due to the fact that mom's other tissues are oxidizing that less than the other classes, so it's more available to the mammary gland and ends up in milk. So that's chapter 1, and to kind of reorient you to the big questions that I'm chasing after, how does all that fat get into the milk, and how does mom ration her lipid reserves? And we'll move on to chapter 2, and that's the big fancy title, but in reality I'm just going to talk about hormones for chapter 2. So chapter 2 is looking at the hormones that can affect the release of fatty acids from blubber, and I'm interested in how that also is related to the milk lipid content. So I spend a lot of time thinking about hormones, but I think probably most people don't, and a little bit of review about what a hormone is and what it does. It's a chemical messenger that travels in the blood, and it's secreted by a particular endocrine organ, and it requires receptors on a target organ before it can cause that enact a change. So once that receptor picks up that chemical messenger in the bloodstream, it can either stimulate or inhibit different metabolic processes dependent on the particular hormone. The particular hormones I'm going to talk about today are cortisol, growth hormone, and insulin. Cortisol, you've probably heard of, it's a general stress hormone. It does do lots of other things, but one of its capabilities is to increase lipolysis, so increasing the burning of fat. Growth hormone, among its other many actions, has also been shown to facilitate lipolysis, so facilitate burning fat. Now, insulin, you've probably heard of in relation to glucose metabolism, but insulin also has effects on lipid metabolism, and it inhibits lipid breakdown. So it's sort of like the brakes on lipolysis. So lots of insulin, you burn less fat. Lots of growth hormone, lots of cortisol, you burn more fat. So I'm interested in how do these different hormones affect lipid breakdown in elephant seals. So to do this, again, we went out to Año Nuevo, and for this chapter I had samples from both 2005 and 2010, so I was able to have a pretty decent sample size. And we have milk samples, blood samples, again we measured mass, and the body composition, so how much lipid mom has in her whole body. And then I took these samples to the lab, and for the milk, I was able to extract all the lipids and get a percent lipid composition. So after breathing lots and lots of ether, I ended up with a percent lipid composition of the milk sample. And then for the hormones, some of them I did at Sonoma State in Dan Crocker's lab, my committee member and collaborator, and some of them I was able to do here at UC Santa Cruz. And I also measured metabolites in the blood, so the free fatty acids and the TAG in the blood as well. So that was all the lab work. And then I have results. So for this panel, it's going to start up here. This is insulin, early, late lactation, early, late molt. And we see a large drop in insulin across lactation, but we've got stable levels in the molt. And then down here, we've got cortisol, and cortisol grows up over both early and late lactation, or from early to late lactation and from early to late molt. And in fact, the magnitude of the increase is much larger in the molt. So we've got decreasing insulin in lactation and increasing molt in both lactation and molt. Oh yeah, so I think I meant to mention that I also measured some of these in molting animals who are fasting but not lactating. So the molting animals are representing a fasting animal but not lactating. So that's kind of the comparative aspect, where we've got lactating animals who are creating milk and fasting, and molting animals who are fasting but not lactating. So I meant to mention that for the last slide. All right, so growth hormone. we've got a slight increase across lactation, but stable levels again in the molt. Now, those are our baseline values for the hormones. When I looked at what was going on in the blood, we've got a slight increase in triglycerides in lactation, again, stable in the molt, and a dramatic increase in our free fatty acids in the blood, and it's stable over the molt. And as far as mom's entire adipose stores, how much blubber does mom have? Over lactation, she depletes more of her blubber stores versus over the molt fast. So lactating animals are depleting much more of their lipid stores, which makes some sense. She's making a lot of lipid-rich milk. So those were all baseline values, but I really wanted to know how do all of these variables interact. So how are hormones related to our free fatty acid levels in the blood, right? Because that's what I want to know. How is it affecting releasing these into the blood? So I used a linear mixed model, and I looked at the effect of growth hormone, cortisol, and insulin on free fatty acid levels. And what I found is in lactating females, the increase in cortisol and the decrease in insulin significantly affected circulating free fatty acid levels. But growth hormone was not related. And in the molting females, I found only cortisol affected the free fatty acid levels. So insulin was not involved in the molting animals. And I was also interested in how does this affect milk. So these hormones have a relationship to milk lipid content. So I did something similar, and I looked at milk lipid content in relation to growth hormone, cortisol, and insulin. And similar to the free fatty acid results, I also found that increasing cortisol and decreasing insulin are related to milk lipid content. And there's just a few more variables I was interested in how they kind of fit into this whole picture. So I've got those adiposity measures, I've got free fatty acids in the blood, TAG in the blood, and the mass of the female overall, and how do those affect milk lipid. And I found that adiposity, so how much her lipid stores, and the circulating free fatty acid are both strongly significant predictors of milk lipid content. And I also found TAG and mass are also involved in predicting milk lipid content. So I think this argues for regulation at the level of lipid release. So you've got the stores of blubber available are predicting milk lipid content. You've got hormones that are affecting the release of milk lipid of blubber, and that's affecting milk lipid content. So I think this argues for the regulation of milk lipid content at the level of release from the blubber stores. So put this all in a little bit of a pictogram form for you. In lactation, we've got our blubber, our adipocytes. Free fatty acid is released, it increases in the blood, and is picked up by the mammary gland to make milk. And what I found increased cortisol and decreased insulin significantly affect this release of free fatty acids. And then when I looked at milk lipid content, I saw cortisol, insulin, free fatty acid, TAG, mass, and adiposity. All of those are also significantly related to milk lipid content. Now, if you contrast that with our molting animals, so these molting animals are fasting but not lactating. And again, they're releasing free fatty acids from the blubber, but they're not releasing nearly as much. And I found that cortisol was related, but insulin was not related to driving this release from the blubber stores. So insulin seems to be the different factor between lactating and molting seals. And it seems that we're basically removing the brakes from lipolysis by decreasing insulin is really important to lactation, whereas molting seals, cortisol seems to be driving the release of lipid. So take-home message number one is that insulin is a differential regulator of lipid metabolism in lactating seals, and that there is evidence for regulation of milk lipid content at the level of the release from the blubber stores. And the last thing is that growth hormone is not nearly involved in lipolysis as expected. I actually thought that was going to be like the home run when I started. Growth hormone is totally the answer. It's doing everything. And it's not doing anything that I can measure. So that was interesting. All right, so that's chapter 2. Moving on to chapter 3. And again, our big questions, how does all that fat get into the milk? And how does mom ration her lipid reserve? So for this chapter, I am investigating hormone-sensitive lipase and adipose triglyceride lipase Those are these two enzymes that cut up fat and lipid. So this is this portion right here. Again, they're releasing those stored tag molecules from our adipocyte, or the sealed adipocyte, ours too. In order to do this, again, on nuevo, some of these seals have donated samples to several chapters, but I've got milk and blubber samples, and for the molting animals, I only have blubber samples, obviously, and I've got blood, mass, and body composition. And I wanted to see the activity of these enzymes in the blubber and how they're related to milk lipid content. So I went to the lab, and again, I've got my breathing lots of ether and calculating how much lipid is in that milk sample. And then the lab activities for the enzymes was quite an adventure. So I had no idea how to do this, and I started doing a little research, and no one that had ever done it in elephant seals, and I started looking around, and I discovered that there was a researcher over the hill at Stanford who does this regularly in the clinical literature for molecular biology. And so I called him up and said, hey, I want to do this in elephant seals, but I don't know how to do it. He said, yeah, come over here and we'll show you how. So that was awesome. I thought it'll be awesome. I'll just go over there and just whip that up. And it took a lot longer than I thought it would. A lot of quality control to make sure these assays that are developed for mice, that we felt comfortable in using them for our seal samples. But in the end, what we ended up measuring is the activity of these enzymes. And so when I talk about activity, I'm talking about a measure of what these enzymes actually do. So in a super, super brief, simplified form, you take a sample of blubber that has these enzymes, and you put it in a tube, and you put it with extra triglyceride, because that's its job, right, to cut off those fatty acids. And then you let it do its thing, swirl it around, and it cuts off fatty acids, and then you see how much fatty acid did it cut off. And that tells you how active that enzyme is, how much of its job is it really doing. So I have that measurement for both those enzymes. And then I also did a measurement that looks at the presence of these proteins. They're enzymes, they're proteins, and so they're expressed in a cell. So you can look at like the amount of expression, so how much protein is actually there. And this involves separating them by gel electrophoresis, which will separate them by size, and then putting antibodies on there that have fluorescent little markers that light up when it sticks to that particular molecule. So it will recognize that molecule and light up, and when it works right, you get these really pretty pictures, and sometimes it doesn't always work right, but that's what it looks like when it works well. So I also have a measurement of how much of these proteins were expressed. And then I also have my blood metabolite levels, again, our free fatty acids and our tag levels. All right, so jumping into the results. So hormone-sensitive lipase, first off. This is one of those Western blots. Over here is a marker. So that just tells us the size of the different molecules. This is a positive control. It's actually mouse fat. So a lot of these assays are developed for mice, and I wanted to make sure the assays were working, and I also wanted to be able to compare that to the elephant cell results I was getting. So here, this works great. This is just what you would expect. That's wonderful. And then we've got our seal samples over here, and at first, oh, there's bands. That's great, but they're the wrong size. So they're down here, and that's not what you would expect. And after doing a special experiment to evaluate how well those antibodies were recognizing the molecules, and I'll spare you the details on that little experiment, we basically determined that we're not able to measure hormone-sensitive lipase with any kind of confidence using the antibodies that are available to us. But then that actually made sense when we started looking at the activity data. So again, this is just presence of the molecule. And then looking at the activity, so what that enzyme's actually doing, how much lipid is it cutting out, we found that there was really low levels. So you think that an animal that is heavily dependent on mobilizing lipid, everybody for years has thought hormone-sensitive lipase is going to be the thing, that's what it's doing. I thought, oh, there's going to be just sky-high levels. It's actually really low. And so when compared to the mouse samples, it's like 7 to 15% of what's happening in a mouse sample. So like, oh, okay, well, we're not able to detect it in the Western blot, and there's not very much activity, so maybe hormone-sensitive lipase is not the thing. So we moved on to ATGL. And first thing we did one of these Western blots. This is our mouse sample. This is the marker. And these are seal samples. Yay! We're seeing them. They're great. They're great size. This is wonderful. I'm so excited. We're going to proceed with ATGL. And when I was looking at these, so here's a marker. Here's a mouse sample. We've got early lactating seal, late lactating, early, late. And you can see very clearly there's a much darker band in the late lactating samples, which means there's more of that protein present. All right, and the really cool thing about this program that I was able to use is that you can quantify the brightness. So again, there's antibodies sticking to this that have little flashing fluorescent lights on them. And you can actually measure how bright that is, which means there's more of that particular protein there. And so I can quantify how much is there. And I'm able to show that in late lactation, there's more expression of this ATGL protein than there is in early lactation, as well as in the late molt. So both across lactation, the expression of this protein increases. But then I was looking at the activity of this enzyme, and I found that the activity is actually very similar. From early to late lactation and to early molt, all of these are extremely similar. So even though there's more protein around, it's not doing any more in the different stages. This result with the late molt, so that's a little bit interesting. I'm not really sure how to explain that, but I did see in the late molt that there is higher activity. But in late lactation, we have lower activity, even though there's much more of the protein being expressed. And what I was ultimately interested in is how do the activity of these enzymes affect milk lipid content and lipid release. So then I looked at the relationship to milk lipid content and to circulating free fatty acids, and I didn't find any relationship, which was a bit disappointing. But yeah, so I wasn't able to detect a relationship between the activity or the presence of these proteins to milk lipid content or free fatty acids. So I started thinking about what might be causing this. How come I'm not measuring any relationship between these? And as I'm learning with molecular biology, there's always some other little molecule that swoops in and has something really important to do with what you want to explain. And so there's a couple different options of things that I didn't measure that might be more fine-scale regulation of these biochemical processes. And not to spend too much time in the biochemistry, but there is a molecule that can actually block the access of these enzymes to the lipid droplet. And so if an animal is expressing lots of those, even if you've got lots of enzyme, it can't get to the lipid droplet. So in other studies, they're finding that there's this molecule called paralipin. There's also other molecules, for example, one called CGI58, that is necessary to make ATGL do its job. So you've got ATGL, but if it doesn't have its sort of helper, it may not be doing a very good job at doing its job. All right, so then a particular organism could upregulate or downregulate that, helper protein, which could affect your activity. So there's a couple of different fine-scale things that may be happening here that I wasn't able to detect with my kind of coarse first run at measuring this in an elephant seal. And again, a lot of these would probably be better investigated in intact cells. So I took a blubber sample and I froze it and then I ground it up and did this. But if you could culture a cell and actually see this sort of in real time in an actual functioning cell, you might be able to get a better handle on the sort of intricacies of how these different helper molecules are affecting this process. So one more result slide that was pretty interesting. So I actually found that seems to be a bit of a scaling effect, where we've got larger seals have lower ATGL activity than the smaller seals. And this follows with previous research that has shown larger seals have lower metabolic demands than smaller seals. So a bit of a scaling effect. So that was an interesting finding in that larger seals have lower activity than smaller seals. So our take-home messages for chapter 3 is that hormone-sensitive lipase is much less important than previously thought. So that was actually a really interesting finding. I had no intention of measuring ATGL when I first started this. I thought hormone-sensitive lipase is going to be super sky high, it'll be a home run, it'll be great. It's not. So that's an interesting finding. And in fact, ATGL seems to be the more important lipolytic enzyme in fasting elephant seals. And the other take-home message is that there's probably some more fine-scale regulation happening here that that wasn't really able to detect with this first pass, and there's probably some expression of chaperone proteins, or there's other signaling molecules that could be involved that are regulating how these different enzymes work from early to late lactation. Even though a seal is making more of the enzyme, there's other ways to regulate how it actually works. All right, so to wrap things up, my big questions I've been harping on all day: how does all that fat get into the milk, and did I actually answer any of that? First off, I was able to show that the regulation of the lipid mobilization strongly impacts milk lipid. So at the supply side of the whole equation, how much lipid is available to the mammary gland, it seems to significantly impact how much ends up in the milk. I was also able to show that the decrease in insulin is really important to liberating fat in lactation. So removing the brakes of insulin enables these lactating seals who have to create huge amounts of milk and liberate huge amounts of their lipid stores. The removal of insulin is very important to that. And as far as future directions, I think that there could be some interesting work looking at some of those specific transport proteins into the mammary gland. If there's one of those that is selective for different particular types of fatty acids, that would be really interesting to look at, as well as actually measuring the oxidation of these specific fatty acids. So I kind of inferred oxidation, kind of looking at a big picture, but if you were actually able to measure the oxidation of these specific fatty acids, that would further inform, how much is mom actually using from her own tissues versus what ends up in milk. So I think there's some interesting avenues for that. And then, how does mom ration her lipid reserves? I found that elephant seals mobilize specific fatty acids according to biochemical predictions. So similar to other animals, despite the fact that they're mobilizing huge amounts of these, they're doing it in a similar fashion as other animals that have been measured. And I also found that ATGL is far more important than hormone-sensitive lipase, which is a kind of new and unexpected finding. And I think I was also able to identify a couple of future directions for looking at the regulation of expression of ATGL and the regulation of ATGL activity, which is somebody else's thesis. I think I've done enough lab work for now. But yeah, there's plenty of things to be looked at as far as lipid mobilization and its regulation. So to back up just a little bit and wrap things up, a lot of this talk has been pretty fine scale, pretty biochemical heavy, but I do want to back up and remind everybody that lipid mobilization is super important to elephant seals. It is critical for them to be able to pull off this lactation strategy of foraging far out at sea, exploiting this really this food resource that is so far from land, and being able to continue to come to land and breed, where they're tied to. They have to be able to be really good at accumulating fat and then mobilizing it really quickly. And it's important not only for mom to be able to do this, for her own health, to mobilize all this lipid, and then she's got to ration that carefully so that when she goes back out, she can do this next year. She's got to make sure she mobilizes just enough, gets junior just fat enough, and then has enough to get out to sea and recoup those losses to do this whole thing again. And again, it's also super important to the pup to have this lipid, to be able to be fat enough so that then it can develop the diving capabilities to go out and forage effectively. So even though it's sort of a lot of fine scale, I think that it's really important still even from a broad perspective that lipid mobilization is one of these critical aspects to elephant seals occupying this particular niche of foraging far away and being tied to land and pulling that off effectively. So hopefully you think the same thing. So with that, I'm going to do a couple acknowledgments, and I'm going to try to keep this brief. I know the acknowledgments part gets a little bit long sometimes. So, cool. I want to thank the Costa Lab, and there's tons of people. The Costa Lab is notorious for being giant, and so there's just tons of people to thank. Everyone has been amazing to me over the years and really supportive and helpful, even though I study things that none of them have very much knowledge about. They're still always supportive and helpful, particularly Corey Champagne. Thank you so much. for holding my hand through all my daily mini catastrophes. Guido McDonald, who's not here any longer, but is one of my heroes and just an amazing role model for how I want to be as a scientist. So she's been super helpful, and I just really look up to her so much. Luis, who's not here either, and Autumn Lynn, who is here. You guys have just been there for me every single day through everything. Thank you for that. And there's tons of people, I can't go through every single one of them, but Costa Lab, you're all amazing. Thank you for everything. And as well, some of my amazing field assistants. There's lots of people and that get up in the morning and ride their bikes here at 5 in the morning in the pouring rain and stand out in the blasting wind with me for 5 hours and do it every single day, and they do it cheerfully, and I couldn't do it without you. So that's amazing. Thank you so much to my amazing field help. The lab in Palo Alto, Dr. Kramer and Wenjun, who are here, as well as Shalja, Masami, and Victor. I was pretty clueless when I showed up, and thank you for your patience and walking me through it, and I learned so much from you, and it's just been really amazing and allowing me to utilize your resources, so thank you for that. As well, I've got a huge list of friends, both here in Santa Cruz, as well as my friends from New York who came from undergrad, so it means a lot that you guys came. Sorry. My cohort from 2007, and it's been really good to have people to go through all those steps of what do we do now? Now we have to pass our comps. What do I do? So I made it. You can do it too. So thank you for that. I also want to thank the Rangers on Nuevo and Pat Morris. It's just really incredible to have a resource so close and to be able to learn so much and for it to be so accessible. So that's amazing, and thank you for facilitating all of that. I want to thank my family. My mom is here. Unfortunately, my brother and his wife were coming, but there's a lot of snow between here and Idaho, so they didn't make it, which makes me really sad, but I really appreciate their help and their thoughtfulness in trying to come. Mom, I do want to thank you for remaining continually calm, even though I call you up and tell you that I'm going to Antarctica or Galapagos, and I'll see you in 6 months. I'm sure it will be fine, and she is supportive and handles it no matter what crazy crap I tell you. So thank you for that. And then my other brothers, Adam and Nathan, and then my aunt, who was supportive when I decided to run away to New York and start this whole process. So I want to thank my family for that. And then my committee, Dan Crocker, thank you so much for all the years of being really supportive and there for me. Kathy Debier and Terry and my main advisor, Dan Costa, thank you guys so much for all of your help through this process, as well as being supportive of the fact that I wanted to go measure this crazy enzyme that nobody knew anything about and letting me go and chase that down. So thank you for that. And also for letting me take random projects in Antarctica and Galapagos, even though it had nothing to do with my topic. So they're like, yeah, sure, that's fine, go ahead. So thank you for being patient with that, and thank you, Dan, for all of the opportunities. Being in your lab, I've just been able to do so many things and meet so many people, and that's really fantastic, so thank you for that. The EEV staff, Susan, Debbie, Kathy, and Maria here, helping through all those logistical challenges and struggles. Thank you for that. And I wanted to thank Pete Ramonde also for, I've been able to do a lot of teaching here, which has been a really amazing opportunity, and I've learned so much. So I wanted to say thank you for that opportunity. And as well as Rita, and I don't know if she's here, but she's been a really great mentor to me and been really supportive, and I wanted to thank her for that. It meant a lot to me. And with that, I am done, and I would be happy to take questions. Thank you. Burning lipid metabolism questions, I know you've got rolling around in your head. Yeah, Robin?
Hey, very fun. I love talking about fat. Really exciting. I did wonder about how much variation is there in the body composition of the females and the bees, and how do they regulate? Like, is it the kind of thing where once I get to this % fat, I gotta leave and leave this pup, or is it a percentage of body mass, or how do you envision the enzymes might be involved in that process?
I think that the signal leave is probably more of a protein catabolism, but when they kind of get to the end of their, because they do, even though they're predominantly catabolizing fat, I think the kind of limiting factor is probably the protein. So I think there's I can't remember the exact variation at the end, but I think the signal to leave is probably more of like, I don't have any more protein. I still have a little bit of fat to go, but so the signal to leave, I think, is more protein based.
And what about leptin in all of this?
So I have not looked at leptin. I think there have been a couple of studies who have attempted to measure leptin, and I don't think they found anything definitive in elephant seals. And so I don't know if that's an assay problem as far as being, because that's always one kind of question in things like if it's not developed for a seal. So I don't think there's any definitive leptin. If you guys know of any off the top of your head.
You measure it, but it's not driving.
It's not driving anything, yeah. So it doesn't seem to be important in the equation. Terry, did you have a question?
Yeah, it was fascinating. I really, really enjoyed this. And the thing I found most fascinating is that you started off like everyone else, where it's like these pups and they're really small, they're getting bigger and bigger, and mom's just giving and giving and giving. But your story ended up being one of conservation by mom, that at all costs preserve yourself, whether it's at the molecular level or the particular fatty acids that are going to go to the pup. So I started to wonder, if you were to look at this say, on southern elephant seals, or different kinds of environments where thermally the mom is being challenged, either hotter or colder, do you think that would change your story a bit? Just because it's such a, it seems like she's just defending herself while she's trying to do this amazing thing with the pup.
I think it might have a bit of an impact. Yeah, I think, but I'm not sure how much, because I think there seems to be quite a bit of the medium chain MUFA, which are kind of the ones that go outside. So those are the highest, the largest class, and so there might be more of those required for a colder environment to be on the outside. So that might change the interplay a little bit. And yeah, I don't know, because I think a lot of it does seem to be driven by some of these kind of basic biochemical processes of oxidation, and that's the way chain lengths are. But then if they're depositing more in that outer layer, so I think it might have somewhat of effect. I don't know if it would be super dramatic though. But yeah, it's just guess. I'm not sure. In the back.
So how efficient is the weight transfer from the mom to the pup? And then is that more or less efficient than an animal that isn't fasting?
All right. So efficiency, it's like 30 or 40%. This is Dan Crocker's thesis, right? The efficiency. It's like 30 or 40%.
It's metabolism and rest.
What's that?
So 30 or 40% is metabolism and rest.
Yeah. So the rest goes in milk. So that as far as mom or efficiency transfer.
As far as of mom's losses.
Yeah, so if mom's losses, 60% goes to milk energy, which is, well, it's also kind of interesting to compare to other lactating animals, because most other animals aren't fasting, but that's pretty darn efficient. Anybody else? Yeah.
I've been wondering about the milk ratio, changing the milk fat ratio changes. How much of that could be just limited osmotic analysis, perceiving fats? Is there just less water available?
There is less water. Yeah, there is less water in the milk, but it's just it's a percent composition across the whole sample. So yeah, there's less water, but then there's just more milk to kind of make up for the difference. So yeah, it probably is part of that, is that they're not drinking anything either, and the milk is sort of just continuing to put fat in it, kind of can replace that water. Anybody else?
All right. Well, I think we can formally call her Dr. Melinda Fowler, and I'm going to bring cakes and champagne, and we can toast her. But I forgot to, in the introduction, I forgot to make a point that amongst all the other things that Melinda did, she actually was one of the people who came in with a STEPS fellowship, which is quite a prestigious thing to come in with. She also not only got a presidential dissertation fellowship, she also got the ARCS fellowship. So she, I should have, as I was sitting there, I remembered I forgot to make that point, but it was on her slide. So Anyway, great job, and you guys can enjoy yourself. I'm going to go get the refreshments.
It's nice work, Melinda.
Thank you.